Hi everybody, so today we're going to do our plant part identification and leaf morphology lab activity. So this is to reinforce the typical life cycle of a monocot. As you see here on this paper, the radicle and plumule will be the first things to come out, the root and the shoot. Next, we're going to have the radicle growing downward, and then we're going to have some secondary roots branching out, which is the growth and branching phase of a monocot. The next stage that we have is adventitious roots forming at the shoot axis, just at or below the soil surface. And these adventitious roots act as an anchor to further support the plant. Here we see a monocot diagram. On the right hand side, you will see the diagram of the corn growing, because corn is a prime example of a monocot. We have the primary root growing down, along with secondary roots, a true leaf, and the cotyledon being shown. The seed at the top means for each part of the seed. I will show you those here momentarily. This here is my corn seedling. The blue color is a fungicide treatment that is applied to field corn. The white kernel is sweet corn, and that blue kernel is the same. Both of these have not been germinated yet. They've just been imbibing in water. I'm taking a razor blade, and I'm going to use this to cut my samples. Next, we're going to compare the seedling I have to the monocot diagram to try to identify these different parts. What I am pointing to here is the true leaf and coleoptile region. The next that I'm going to be pointing out is going to be the primary root and secondary roots that are emerging. I'm also taking the razor blade to cut this kernel in half. What I want to see is the cotyledon. If you look closely, you can see that little dark spot, and that's going to be the cotyledon. The next thing we're going to talk about is the typical life cycle of a dicot. We can see here the first step is that the radicle emerges. The next step is the hypocotyl and cotyledons are emerging. The hypocotyl just means anything below the cotyledons. Our next step is apical meristem grows upwards, and so it's going to straighten itself out. And our last step is that the apical meristem continues to grow and the leaves flourish. I am removing my bean seedling. Bean is the most common dicotyledon used for an example, because you can see the first two true leaves and the two cotyledons flawlessly. So the white seed that you see there is a soybean, and then those brown seeds are bush bean types. And so we see the hypocotyledon, we see the roots, I'm going to take the razor blade again and cut the seeds in half, and I will also cut the germinating seedling in half. What you see here are two cotyledons and the first two true leaves. And so I'm going to separate that. What's on my fingernail are those leaves that are going to emerge, and the cotyledons is basically what was in the seed. So what we're moving into is the lab assignment. This is dealing more with leaf morphology and the identification of leaf parts. And so this is what I handed out during my lab, and it's also available on iLearn in your lab section. So what we're going to do is that we're going to have 4 specimens, we're going to trace them, we're going to label the different parts, and we're going to discuss in the discussion post for your assignment what was the difficult part about this assignment. In my recorded lecture, I mentioned that the diagram that was listed did not truly encapsulate what the blade of a leaf is. And so I created my own, and on the left hand side you see the word blade, and it's the whole entire leaf itself. The apex is still the top, the margin is still the outer ring, the veinlet, midrib or a midvein, and then of course the base. These are the samples I collected from outside of our building. The marijuana looking leaf is actually called a lupine. We have a red bud, an oak leaf hydrangea, and an unknown leaf. I call it unknown because as I was walking around I found it on the sidewalk, decided it looked great, and it is a part of the collection now. You will see me take each of these leaves and complete the activity. I just wanted to be able to show you all 4 before I started working. The first step is to take your leaf specimen and outline it. I am not going to fast forward these clips because if you are following along, I want to give you the time to try to draw what I am tracing. And this is the oak leaf hydrangea that is up first. And so I'm not skipping anything, I'm going over it all. Our next step is to label the basic parts of what we're identifying. We're going to label the base, the margin, as well as the apex. The first thing we're going to identify is the base. Here is the handout I gave out in lab. It has 4 common base types, and so we're going to compare that to our oak leaf hydrangea. Comparing the base, I determined that the rounded base is the most similar to the oak leaf hydrangea. The next part of the leaf to identify is the apex. These are 4 common apex types, and what I'm going to do is take the leaf and hold it to the sheet and try to identify which one is most similar to our apex. After comparing the apex to all the ones on the sheet, I determined that our apex for the oak leaf hydrangea is the mucronoliate apex. The next step is to identify the margin. These 6 margins are the most common, but there is plenty more than what is listed here. The thing with margins is that there can be more than one, and so we're going to try to identify which one our oak leaf hydrangea has. As you notice here, there are some teeth on the margin, but you also see the different shapes in our margin. It is not one beautiful round oval. We got some cuts going on, some roundedness going on, okay? So I considered this a serrated, parted margin. This is showing my trace job, the leaf, and how I labeled the apex, margin, and the base. Please feel free to pause to write this down. We're going to be moving on to our next specimen. Our next specimen is the lupin or lupine leaf. I'm going to do the same thing. I'm going to trace, and you will notice that I do struggle because there are multiple leaflets for this specimen. And so that will come in handy in just a few moments. So as you notice, this leaf looks different than our oak leaf hydrangea. And this actually is due to a division type. It is a palmate division, which means it's a compound leaf, and each one of those little fingers is a leaflet. And so you see me here comparing, and I'm going to write it at the top. It's palmately compound. I'm going to label the apex next. I'm going ahead and filling out the rest of the labeling, so we're going to also do the margin and the base. Once again, I'm going to show the apex sheet, and you will see me tape the specimen, put it over the sheet, and try to make a correct match. The apex for this specimen is called a ristate, and it fitted very well, if not perfect. Next, moving on to the margin. It's smooth around the edges. There's no texture. And so, whenever you try to identify the margin of a compound leaf, you look at each leaflet. So that's an entire margin. Next, we're going to try to identify the base. And just like with the margin, we have to look at the base of the leaflets. So although it is palmately compound, each leaflet has to have one of these types attached to its petiole. After inspecting each leaflet, I determined that the base is called oblique, which means that one side comes down a little bit farther than the other. So this is the end result for our lupine specimen. Please take the time to pause and write these down, or make notes, or just observe. Our next specimen is a redbud. And so I'm going to trace it again and go through the steps of labeling the apex, margin, and the base. So we're going to look at the shape. So a red bud is typically a cordate shape, which means heart shape. And so I'm going to match it up, and it's going to be cordate, and I'm going to write it at the top. So next I'm moving on to bases. So you would think if it's a cordate shape, it should have a cordate base. However, if you look at this leaf, this one does not. but this can also vary leaf to leaf. Next, we're going to move on to margin. It's smooth, there's no parts in it, it's going to be an entire margin. Next, moving on to the apex, and so once again, you've seen this sheet already so many times. We're going to try and pick which one matches it the best. I've determined that the best apex for this specimen is also going to be the mucornoliate apex. Once again, I have this picture here that shows the redbud leaf, the parts that I determined to be identified as. Please take the time to pause or to consider or even decide something different. Last but not least is our unknown specimen. It's going to get the same treatment, it's going to be traced, it's going to be prepared for labeling. We will get to that in a moment. So we are going to determine the apex of this unknown specimen's leaf. I'm going to take the leaf, slide it over each of these options, and see which one best fits our tip. I determined that our apex is the acute apex because it's a cute little tip. Next, we're moving on to margin. It's smooth, no teeth, there's no deviations through it, so it's an entire margin. Here are the bases again, and I'm trying to decide which one is which. I determined the base is rounded. Next, I'm going to figure out the shape. Not all leaves can fit into these different shapes, but this one could very well fit into it. So I'm going to take it and I'm going to compare it to each shape on this list. I'm going to determine is an elliptic shape because it is not oval, it's not the same width all the way around, it's more elliptic. Once again, here is the unknown specimen with the determined identification of the apex margin, base, and in the shape. Please take the time to pause or to consider or even just come up with a really good argument to why this is stupid. I want to take a moment to explain to you why this is important. Being able to identify the parts of the leaf and understand these basic types of margins, bases, apexes, and shapes can help you aid in understanding your plants. So if you are trying to identify the weeds in your field, this is really good to know because there's a lot of handbooks that will determine if a certain species has serrated or if it's certain shapes. And it's also really good for conservation efforts.